Hello everyone and welcome to my video lecture. Today I will provide you a background and overview of enzyme purifications. Enzyme purification simply describes a variety of methods available to isolate a specific type of protein or enzyme from a cell extract, which is a mixture of proteins. The goal of this is to isolate the target protein without deactivating it. Deactivating a protein means that it is no longer functional. Sometimes this can be revived, but other times it cannot. We will go into greater detail of each of these techniques, but below is a handy overview of what each technique accomplishes. Ultimately, as a protein becomes more pure, its activity should increase. It is important to note that many times these techniques will be used in combination, by starting with one method and then using other until the protein can be as pure as possible. Let's talk about the techniques used for enzyme purification. First method is centrifuge. Here, separation takes place on the basis of density. Second method is salting out. Here, separation takes place on the basis of solubility. Third is dialysis. Here, separation takes place on the basis of size. And fourth method is column chromatography. This method is divided into three different types of techniques. First is gel filtration. Also called size exclusion method. Here, separation takes place on the basis of size. Second method is ion exchange chromatography. Here, separation takes place on the basis of charge. Third is affinity chromatography. Here, separation takes place on the basis of affinity between sample and ligand. So these are the methods used for enzyme purification. Let's talk about centrifugation, which is the first method for enzyme purification. As we learned in our overview of protein purification methods, centrifugation is the separation of proteins by density from a cell extract. When designing a purification scheme, this is generally the first technique that is used. The process of centrifuging simply relies on spinning a tube at very high speed and will ultimately pull the higher density proteins to form a pellet in the bottom of the tube. You can see the diagram. By altering runtime and speed, different fractions can be collected which will each hold a different protein makeup. We can extract protein from the bottom of the test tube, or this is called as pellet. So this is all about the protein purification methods. In next video, I will tell you methods in detail. Thank you.